Hello and welcome to online lecture 8. In this unit we are going to discuss DNA mutations and disease, and we're going to focus on a case that involves a disease called Huntington's chorea, and we're going to use the same software, the lec8 software, that we used in the DNA microarray lab that we did on breast cancer. We're actually going to do the case on Huntington's chorea in class on Wednesday, November 6th. And it would be really wonderful if those of you who have laptops can bring those to class with you that day. And if you can go ahead and install the lec8 software that we used in lab, that would be really helpful also. So the purpose of today's lecture is to give you a little bit of background on Huntington's disease and on the two molecular techniques that are used within the lec8 software. First, let's start with a little bit of background about Huntington's disease. Huntington's disease is a genetic neurological disorder, so that means it's inherited and it affects the nervous system. Generally speaking, symptoms appear around age 40 and progress gradually from subtle abnormalities in movement to dementia and eventually a loss of complete motor control. It is uncurable, although medication can mask some of the symptoms. And some famous people, such as Woody Guthrie, have been victims of this disease, and he ended his days in a mental institution. The biology of Huntington's disease is only partially understood. It's caused by mutations in what's called the Huntington gene, and we'll look at those mutations in detail in the case. And it's known that these mutations can lead to the buildup of damaging proteins in brain cells, that the disease actually leads to the death and deterioration of nerve cells in the brain. And if you look at this slide over here, the photograph, on the right hand side you can see a slice through a healthy brain, and on the left hand side you can see a brain that's been infected by Huntington's, and these black areas are showing areas where the brain tissue has decayed. Huntington's chorea is characterized by a group of movements that are called chorea, and these are basically involuntary abnormal muscle movements. And in this video you can see a woman who's afflicted with Huntington's chorea that has progressed fairly significantly, but you can see she's also still able to have some function. So you can see she's able to still move with an intention and appears to have some ability to understand what's going on. Symptoms of the disease can vary between individuals and even among affected members of the same family, but the disease usually progresses fairly predictably. The earliest symptoms are often subtle problems with mood or cognition. A general lack of coordination and an unsteady gait often follows. And then as the disease advances, you see the uncoordinated, jerky body movements we just saw in the previous video. And along with this, a decline in mental abilities and behavioral and psychiatric problems. The physical abilities are usually gradually degraded until a coordinated movement becomes very difficult. And generally speaking, the mental abilities decline, eventually resulting in dementia. And there are a lot of other complications like pneumonia and heart disease or physical injuries from falls that reduce the life expectancy to, generally speaking, around 20 years after the symptoms begin. And as I said earlier, there's no cure for Huntington's disease. And generally speaking, full-time care is required in the later stages of life. So there are drugs that can relieve the symptoms of the disease, but none of them actually represent a cure. And this disease, as I said before, is neurological, and it's caused by an autosomal dominant mutation in this Huntington gene. And although I mentioned previously that the mechanism which causes the disease is not entirely known, What is known is that there is an expansion of a 3 base pair repeat, a triplet repeat that consists of a cytosine, an adenine, and a guanine. And the number of those copies that an individual has in Huntington, so the repeats, basically results in a different muted form of the protein, which gradually damages the cell. This slide is showing a microscopic image of some neurons, and the blue neurons are the unaffected ones. The yellow ones are actually showing what are called these nuclear inclusions, which occur as part of the disease process. So they're thought to result from these different number of repeats in that CAG triplet that I just mentioned. But again, exactly the relationship between the number of repeats and the form of the Huntington's gene and its physiological effects is not completely understood. And our case is going to focus on genetic testing for the disease and family members of people who've been affected. And genetic testing can be performed at any stage of development, even before the onset of symptoms. This obviously raises some ethical debates. How old is a person considered mature enough to choose testing? Do parents have the right to have their children testing? And how should the results be managed? So we're going to explore some of these ethical considerations in this case, as well as actually looking at some of the research methods that would be used to determine whether a person was going to develop Huntington's disease. So the 2 research methods that we're going to focus on are the Southern blot method and the polymerase chain reaction. The Southern blot method was actually described earlier in the lecture on great experiments, and so if you'd like to refer back to that just to help you as you prepare for this case, you would want to go back to the description of the telomerase function and the use of the Southern blot there. I will go through a short video tutorial here which explains the Southern blot process in some general terms. I'll add in some details about that are specific to Huntington's disease, and then I will go over the PCR process. I'm going to do those in a separate lecture, so this is going to conclude part 1 of online lecture 8, and please be sure to watch part 2.